So on to today's speaker, Markus Weiler. Markus is currently the director at the Institute of Hydrology, Albert Ludwigs University in Freiburg, Germany. He's the former chair of Chair of Forest Hydrology, Department of Forest Resources at University of British Columbia, and his doctorate is from ETH Zurich on macropore flow. Markus has been at the forefront of both modeling and measuring macropore and soil flow in using a suite of innovative techniques, many of them focused on isotope tracers. And his talk today, we've invited him in part because one of the important things about the series is that we want to make it be about how people are using isotope hydrology in ways that laser specs make possible and in new innovative ways that really we couldn't have done using the old expensive and, shall we say, slow mass spectrometry. Anyway, so Markus's talk today, I'll let him take the rest of it. Go ahead, Markus. Thank you.
Thank you, Richard. Thanks to all of you inviting me. I'm happy to be with you this morning, this afternoon, this evening, wherever you are on the globe. So I would like to talk about, as you can see, about linking in situ observation of soil water stable isotopes and some focus on water balance estimates, isotope flux in transpiration. So I will go more or less in two parts today. The first one is to introduce you to three more or less new methods to observe stable isotopes, either in a continuous observation in liquid water, measuring isotopes in pore water, or continuous observation of pore water stable isotopes. And then, more or less using those new techniques, I would like to present you two applications where we are using and testing those two methods. One is the potential of pore water stable isotopes for optimization of soil physical parameters, and the second is a coupled soil plant water dynamics during a drought-free wetting transition. So you may already have seen this paper. I just briefly go over it. That's a new method we developed to continuously measure stable isotopes in liquid water using this membrana micromodule. It's just module that has been used a lot in GC analysis, and we just set it up so we have more or less on the one side a stream of nitrogen to the Picaro in this case, but as you know, you can use any kind of laser spec instrument. On the other side, the stream of water that can be stream water, that can be, as you can see in later, a small soil column experiment that's just flowing through that module in the middle, and within the module there's more or less a membrane where the liquid water can transfer to the water vapor. And so what we did there, we tested to see if there's a change in the fractionation factor due to the membrane, and there is one. And this, excuse me.
Markus, are you on the first slide still?
No, I'm on the
first slide.
Huh?
Are you moving the slides? I'm not seeing.
Yeah, I am moving the slides.
Oh, there we are. I'm not sure.
No, I'm 100%. Okay. So I just talked briefly about this new continuous in situ module with that membrana in the middle where we have, as I mentioned already, the vapor and the liquid stream. And we tested more or less the module as the fractionation factor, if it's changing, and what you can see in gray is just fractionation of oxygen 18 or deuterium according to Majube. And from the different modules and different experiment, we can see temperature against the fractionation factor that this is slightly different, but the good answer is this is different from just gas-air interface, but it can be determined, and we can use more or less that mean fractionation factor to predict, using, if you know the temperature at that interface, to predict the isotope concentration in the liquid water. So we did an experiment where we had a column set up where we just changed the initial more or less made just two kind of water types, a lighter and a heavier one. And as you can see in the bottom part, we were just running the laser isotope spec continuously and could nicely see just the transport of the one or the other source of water through the column. And we had more or less measurements at very high temporal resolution, and we did some calculation looking at the difference of water temperature. So we changed the water temperature to see if we can compensate for that. And at the end, we compared the liquid water sample that we took at the same time with the calculation from the vapor. And as you can see, they are all on a nice one-to-one line. The error bars from the vapor measurements are a little bit bigger, but this is mostly because of the fast change in the concentration of the isotopes. If you look at the R squared, that's very high for both, close to 0.998, and we had a relatively small standard error. So we think that this is a relatively cheap and easy way for, with a simple setup, to do continuous liquid water measurements. And we have that no time-consuming analysis of liquid water samples anymore, and we get a comparable precision to the conventional stable isotope analysis using those water samples. The next one, and already Les told you a bit about that last week, is those pore water equilibrium methods. We also use that a lot, and we have more or less set up I think a relatively good lab protocol. We also moved away from the standard Ziploc things that you can see on the left, but now we are using more gas-tight bags that seems to be especially more reliable when we wanted to store the samples not only a day or two, but for a longer time, and that works very well. But the new part we developed is now a continuous in situ observation of the pore water stable isotopes. We wanted to have a robust sensor that we can use for field and lab application to measure continuously in situ water stable isotopes in the saturated as well as in the unsaturated zone. So we just got that published in HESD a couple of months ago. If you have seen it, you can just now relax and you do not need to listen much. If you have not seen it, here's a short run through. So what we did is we developed a small sensor that is roughly 10 millimeters wide. It looks a little bit like a small tensiometer. And in the lower part is a microporous tube. And in that microporous tube, soil water vapor can exchange between the inside of the tube and the soil surrounding it. And then we have developed more or less a system where dry nitrogen source can flow with a specific flow volume into the sensor and will mix there with the saturated soil, with the saturated water vapor coming in from the soil. And that mixing is important in order to prevent any condensation in the tube lines. The other way to do that would be in heating up the sampling tubes, but this is quite cumbersome. So that dilution that we do helps us to prevent those condensation, and so we have a small, robust, inert unit, and we already tested it for several months to years. You can leave it in the soil, and you can just go there again, do some measurement or do some more continuous measurement over time. We put also a lot of time and effort in finding microporous material that works for that is more or less for both conditions, saturated and unsaturated condition, and that is also robust enough to be used in the soil for a longer time. So the whole system looks a little bit like this. So on the right side, there are more or less those different tubes, and the whole thing is connected through a valve block where we more or less electronically can one after the other sensor select the water vapor from each of the sensors. They can be put up in different depths in the soil, so you can just get in situ the water vapor from each of those sensors. It moves back through the lines to your laser spec, and there you can analyze it. We then tested that in the field. That looks a little bit like this. So we have all the different lines. Here in the center part we have the connector and the laser spec in the field to do the measurements. So how does that approach now look like? So we evaluated 4 different methods. The destructive sampling method, as already mentioned before and introduced also last week by Les. We used permanently installed probes where we used an advective. So we extracted the water vapor around the probe, so you more or less suck in water vapor from the soil. And we also tested a diffusive approach where we just had an equilibration between the dry air inside the tube and the saturated water vapor outside. And due to the high permeability of this microporous tube, that equilibration is relatively fast. So this is also other possibilities, especially if the soil is wet or saturated. And the second one, we also just inserted a probe in the soil, so we just pushed in the probe from the top to a certain depth, to 1 meter, for example. Good. So this is how the trace may look like. You see the time in minutes on the bottom, and in the top graph the concentration of water vapor, and in the bottom, in this case, the deuterium values. You see we had a flushing more or less between each measurement that dropped the water vapor in the line, and then we have that stabilization of the measurement to a certain mixing value. In this case, we were around 7,000 ppm, and you can also see that the concentration or the deuterium values are then stabilizing to a certain values, and we can then use that together with the temperature measurements to back calculate the liquid water in the soil. Here's the deuterium oxygen 18 plot for all the different measurements, for all the different methods I showed you before. They all lie relatively good on the local isotopic water line. And just having some information about the evaluation of the whole thing, just highlighted the mean absolute error and the bias of the whole system. For the advective and the diffusive, for the control, so the control is more or less we developed small soil boxes where we put in a known isotopic composition of water, and we had one of the boxes stabilized in terms of the temperature, so the constant temperature, and the other ones we just the temperature fluctuated according to the temperature fluctuation we have in the soil. So we tested that in order how the correction works, and you can see in general we're doing relatively well, and the whole system compares quite well to the destructive control, we so call that. And in general, we're doing a little bit better in relative sense with deuterium as is oxygen 18, but I think we doing fine for all of those. Here's a comparison, for example, of those soil depth profiles. So again, you see deuterium values, 3 different depth profiles with different methods, and the observed depth profile with a destructive sampling every 10 centimeters. I have to say that the destructive sampling was a little bit, let's say, a meter or 2 away from the sensors, since we could not do that otherwise at the same time. Good. To summarize this part, the lab and the field test were very successful. We got comparable precision compared to the destructive analysis of soil water vapor. You can sample roughly 1 sample every 3 to 5 minutes. That's just the time it takes for the flushing, and then until the water vapor arrives at the laser spec. This push-in method is possible. That means more or less you can go out with your isotope sampler, push in that probe to a certain depth, take a measurement, 5 minutes to go deeper and deeper and deeper, so you can more or less sample a soil water profile within a relatively short time. And the possibilities, in my opinion, are relatively wide: water movement in soils and groundwater, root water uptake and redistribution study, or whatever your imagination can be using a sample like this. So I hope I got you a little bit an idea about those two new methods, and I would like to show you two applications where we used the one or the other methods, and what we think is possible in near futures with this approach. Good. So the first one is the potential of pore water stable isotopes for optimization of soil physical parameters and predicting flow and transport in the unsaturated zone. That is work that Matthias Sprenger, a PhD student of mine, is doing within a EFG project in Luxembourg, actually. So the idea was, how can we use additional information to soil water information to inform our models for better prediction of better estimation of soil physical parameters. You know that we have a lot of experience in using depth or time information of soil moisture. Christine Sturm, for example, did a work looking at outflow information of isotopic concentration in lysimeter. Our idea was, does the depth information of pore water stable isotopes provide us additional important information if we want to do inverse modeling of soil hydrological or soil water balance or soil water fluxes? Good. So the research question of the study was, how can the pore water isotope concentration be used as targets for inferring soil hydro properties in the field? And we wanted to compare 3 different optimization approaches to determine the Moellempfang-Genuchten parameters and a transport parameter, in this case a diffusivity parameter, from 3 different study sites. We used a standard model, that's Hydrus 1D, based on Richards equation. For the isotope movement, we used the convection dispersion equation. The upper boundary is our precipitation with the isotope decomposition, ET according to Hargraves, and we used the isotope losses according to a solution uptake that Christine Stump also used in 2012. The lower boundary is a free drainage. We used root water uptake, solute uptake, and the fractionation effects due to soil evaporation are not yet considered, but could be considered in future. For the optimization, we used the shuffle complex evolution algorithm by one, and the objective function is a Kling-Gupta efficiency, so it's a combined efficiency that's just a little bit more stable. Good. So as I mentioned, we used 3 different approaches. The first one we just called isotope profile approach, where we only use the stable isotope profiles, depth profiles in the soil that we sampled at one time, and we only use this, as I'm saying, together with precipitation data and stable isotope data in precipitation to fit the model to that depth profile and to predict the Moellempfang-Genuchten and the transport parameters. The second approach is we call the multi-objective approach, where we used also collected soil moisture and isotope profile information at the same time, and again to optimize or to make the prediction for the Moellempfang-Genuchten and transport parameter. And the third approach is where we have a 2-step approach. First, we fit the model only based on soil moisture data and predicted the Moellempfang-Genuchten parameters, and then in the second round we used the isotope profile information to fit the transport parameters. So the 2-step approach in the only looking at the Moellempfang-Genuchten parameter is nothing else than a standard approach only using soil moisture data. Good. The 3 study sites are a Campisol in Luxembourg, geology is schist. Then there is a Luvisol in close to Freiburg, and a Regosol on a fluvial gravel close to Freiburg as well. So you see the precipitation 600 to 900 millimeters, relatively simple soils, and either on the land use for the meadow or Norway's spruce. So all the sites are on relatively flat ground, so we could assume that the 1D calculation focusing on vertical flow works well for those sites. Good. So here you see an example when you did a little bit of parameter optimization using the regional sensitivity analysis that was introduced by Hornberger and Speer in 1981. And the red line more or less shows if the red line is far away from that diagonal black one, it shows you how well a certain parameter is identified. And this is more or less for the two different soil horizons, soil horizon 1 and 2 that we used in that. And you can see that the information that the optimization of a parameter for most of them works relatively well. When we compare the different approaches, that's for one of the site as an example. As you see the isotope profile, multi-objective and the two-step horizon, then we can see that the multi-objective seems to give us the best sensitivity from all of the approaches, and but also with the other approaches we can determine the parameters quite successfully. This is just an illustration what will be the results. For example, if you now have that fitted model and you run it in this example for 2 years, color coding is the deuterium concentration in the soil from 0 to 1 meter depth, and you see due to the variation in the input we have the different deuterium concentration moving slowly into the soil, but also partly then more or less diminishing, and that diminishing is mostly a result of the evapotranspiration, the root water uptake, so a certain higher concentration or certain heavier or lighter water will be uptaken, and only under certain condition will the water be able to move to a lower part, leave the root zone, and then this is then at the end the groundwater recharge at a certain isotopic composition. Good. If we looking in total for the observation versus simulation for the different approaches, we now compare again for one side how well we do in the left part for the soil moisture, and on the right for the stable isotope profiles. And you can see when you look a little bit in more detail that some of the approaches are doing better for the soil moisture, some certainly because the soil moisture was the first objective to fit to it, some better for the stable isotopes. But it's also clear that if we do not have the stable isotopes information at the beginning, that we often under or over predict the transport behavior. So here's a table summarizing all the Klein-Gupta efficiency for the 3 different approaches and for the 3 different locations, one for the deuterium and one for the soil moisture information. As you see, for some of the approaches and some of the sites, the Klein-Gupta efficiency is relatively similar, but you can also see, for example, that if you only use the isotope profile approach, you may have some issues to predict the water dynamics in the soil due to more or less focus on a certain soil physical parameterization. All in all, we were able for most of the sites to achieve relatively good results, and the results are often relatively stable then. So some of the results you may then get if you look, for example, at the consequences for the water balances. That's an example for the site in Luxembourg. You see on the x-axis time over 3 years, the y-axis cumulative water fluxes in millimeters. So the dashed line is the recharge, the simulated recharge, and the other are the evapotranspiration. And more or less the differences in evapotranspiration or the differences in recharge come from the different parameterization. And if you now compare that for all the different sites, you can see that for some of the sites the differences are quite big. For example, here in Luxembourg, differences in recharge are around 100 millimeter per year in evapotranspiration. This is quite substantial. If you have a total evapotranspiration of around 500 millimeters, for other sites the results are more closely together and the differences are smaller. But still, depending on what information you use, you may get different information back. If you look at, for example, the consequences for transit time, that's often much stronger. We just did two simulations where we just calculated the transit time for more or less looking when the tracer would have applied in the summer or in the winter. And you can see, for example, for the site in Luxembourg, for the summer, the mean transit times for the isotope and the multi-objective approach is around 220 days, whereas if you just use the soil moisture information, it would be 400 days. So the mean transit time would be just doubling. In the winter, the isotope profile approach provides a very short mean transit time, and the transit time between the multi-objective and the two-step approach is relatively similar. On the right, you see more or less for the direct pulse, the breakthrough at the bottom end of our 1 meter deep domain. So you see, first of all, there is quite a differences in transit times between the models, but certainly, as you may know, also depending on the time of the applications. So that dynamic transit time we may see. Here again, the comparison for all different sites, and again, the differences are more or less pronounced, again, depending on the site, depending on the time of the application. Good. So to conclude with that part, we think that the pore water isotope concentration hardly serves as a sole target to simulate soil moisture dynamics or simulate transport processes in the soil. But we think have a combination, and especially a multi-objective approach, gives us satisfying results for simulating water flow and solute transport and provides us the lowest parameter uncertainty. Differences regard to the water balance between the approaches are in the order of 10 to 30%. As I already mentioned, the transit times differ considerably between the different approaches, and also mentioned that the transit time is highly time variant in the unsaturated zone, but this is not new, but often still ignored in larger scale models or studies. Good. Moving to my second example, that's a study, also a DFG project that my PhD student Till Volkmann mostly works on it, together with Christine Haber and Arthur Gessler. They are guys from the plant physiology part, and the work goes on coupled soil plant water dynamics during drought re-wetting transitions. So what is the motivation? We have those dry wet cycles, and there are some predictions that they may change, but independent of that, we want to get a better idea of those potential effects on the water balance, the structure and the functioning of the ecosystems, and we think there is still a significant gap in understanding or in the ability to quantify it, especially those coupled short-time dynamics of soil water and plant water acquisition, and that's where the focus of this work lies. So our first research question was, how do plants affect the soil water balance and infiltration dynamic during drought and re-wetting? That's more or less how the roots, how the structures that are changing on the root water uptake may change the infiltration, maybe due to preferential flow. This is not topic of the whole project, but not in this context now. The second question is, what is the origin of water taken up by plants during dry condition, and how does it change during the re-wetting? And I will show you a couple of examples of that, and how does this relate to root distribution and the physiology of the different plant species. So does different plant species, can they be distinguished, and do they change their acquisition of water in the soil? And third, no, I cannot go further. Could it be that there is a part of the PowerPoint missing?
Do you have another copy locally, Markus?
Yeah. Where can I upload that?
You can open it here if we're missing. That's probably the best thing to do.
Okay,
if you have it locally and easily accessible,
yeah,
you can go to File, Open and Share.
Open chat? No. Transfer.
File, Open and Share, yeah.
Okay. 1 second. Okay, I uploaded it, and it said it worked, but it still don't have any more.
Maybe you only have 38 slides in your presentation.
No, I have 56 slides in my presentation.
Well, the other thing you can do is share your desktop. So you can do, you can do share file.
I didn't check through all of my, okay, share my desktop.
Share file or share my desktop. Just recognize if you have any personal emails here, everyone will get to see them.
Oh, personal emails.
We're looking at your desktop now.
Okay, which side of my desktop? Left or right side?
I'm seeing, I don't see it now. I did see it a second ago. There it is. Here it comes. There you go.
Okay. Probably you see, what do you see now?
Yes, I'm seeing the opening slide.
Only the opening slide?
Yes, it looks like you've maximized the screen. And I'm seeing the opening slide and some content notes. There we go.
So you see this, right?
Yeah, I see
okay,
everything.
now?
There we go, full screen on the performance slide.
Okay, good. I'll just go through. Okay. Okay, we were here, right?
Yes, very good.
Good. Okay, and so our third research question was, can the in-situ laser spectroscopic measurements of soil and transpired water isotopic composition be applied to quantify soil and plant water dynamics non-destructively with a high temporal resolution? Good. So the experimental site is a vineyard terrace in the Kaiserstuhl, so we're very close to important drinking habits in terms of wine. Which again was Louisol, small plot that we planted with beech and oak at a mono and mixed culture. At the end, we had around 4-year young forestry growth and soil physical data at 7 depths and 1 minute interval, and we measured soil water content, metric potential, and temperature. And in addition, we did fine root density distribution measurements with soil cores at, again, different depths, every 10 centimeters. You can see here in the bottom part the root density distribution for the beech oak mixture, and the oak. You see they are quite different, as to be expected. And we then did an artificial drought and rewetting experiment, so we covered the whole part with a roof, so we prevented any precipitation for around 3 weeks. So it's not a complete extreme drought, we just still wanted to have the plants doing fine. And then the sprinkling was done with a drip irrigation tube. We couldn't do a precipitation sprinkler because we also wanted to measure at the same time transpiration of the plants. We used isotopically labeled water with a low intensity of around 5 millimeter per hour, total 40 millimeters of water, and we had no interception and prevented evaporation at the soil surface. Good. So the method we then used is again the continual in-situ monitoring of the soil water stable isotopes that I just introduced before. We measured at 7 depths and we needed approximately 4 minutes per sample, and we did the measurement with the Picaro, and nothing more to the techniques of this. I mentioned that already. For the plants, we used a temporarily closed chamber, so we put a chamber over the different plants. We closed that, measured around 10 minutes per sample, did a Keeling plot estimation of the source water, compared that also to leaf water that we measured simultaneously, and had 3 replicates at each of the site. Good. We used then a linear mixing model based on mass balance equations, where we had the proportional contribution of n sources to certain mixtures, and more or less then did the calculation to examine all the possible combinations for each source contribution, means from different depths, and accept combination that sum to the observed mixture isotopic signature within a certain tolerance. That's more or less the error of our measurement. And for that, we get more or less for the observed source water in the transpired water a depth distribution of sources from each soil depth, where we had again the continuous information from the in-situ soil water isotope measurements. Good. So the sources are our event water, displaced and locally pre-event soil water that we continuously measured. The mixture is the current local soil water at a given depth. And the isotopic event water fraction we determined, and we compared that with the soil physical event water. And secondly, we then calculate the planned water uptake fraction from each depth and compared that with the root density fraction based on our measurements or based on the estimates. Good. So this is how the data looks like. I just take a little bit of time here to explain what you see. Again, this is the beech oak, and the oak on the columns, and then the upper graph shows you the transpired fluxes of actually 2 days we did after the sprinkling event that is highlighted with that 2 dark lines. And then we have the observed isotopic composition in the transpired water. You see we enriched the sprinkling event with relatively high deuterium values to 1,700 per mil. So we had a really strong signal we could work on. And you see the responses are different between the 3 different vegetation. And then in the third row, you see the isotope composition that we measured continuously in the soil water. So again, you see a relatively fast increase of the isotopic composition. Again, that's different. The increase is mostly for the green lines, and the green ones are the upper soil horizon. You see in the bottom left, 5, 10, 20 centimeters. And we couldn't see any response in the lower part, which makes sense because the infiltration front is moving slowly down. And in the bottom line is more or less the observed soil water content dynamics during the sprinkling experiments and afterwards. Good. So I just want to show you now for 9 different time slices that are now marked in with those color codes, the response and the changes that we then see in the different soil depths. The first, you see conventional water content change for the beech, oak, and oak. You can see it was relatively dry at the beginning, and as to be expected, the infiltrated water increased the water content and then decreased a little bit afterwards due to transpiration and due to soil water redistribution. When we look in the bottom part to the observed changes in isotopic composition, you see that you have a relatively fast increase. In some of the sites, the top observations did not see anything at the beginning. We had a little bit of preferential flow close to the surface, but we also did some dye pattern experiments to see if the flow was relatively homogeneous. That was the case in this experiment. So now we calculated the event water fraction. The top graph shows you the soil physical event water fraction based on the soil moisture measurements, and you can see that's what we can expect, a small certain increase of soil moisture content. And the bottom graphs show the isotopic event water fraction based on the mixing model, and we can again see a relatively fast increase, and the event water rise in the top part around to 70, 80, 90%, and below 40 centimeter. Now precipitation water reached a depth below 40 centimeter. But now what's maybe more interesting, because this is now where we can really link the information from the plants and the soil. We now did the calculation of the plant water uptake fraction based on the mixing model, again for the different experiments or for the different tree species. You can see for the beech, the plant water uptake fraction is relatively constant with steps. There is not much variation. For the beech oak, we have a much lower uptake fraction in the topsoil and a higher uptake fraction in the bottom part. And for the oak, this is even more pronounced. If we now compare that with the root density, so that means if the uptake fraction is similar to the distribution of the root density, we should get more or less that proportion relatively close to 0, and this is what we see for the beech, but we do not see this at all for the beech oak mixture and the oak, meaning for those two plant species, the root water uptake is not proportional to the root density. This we can also see if we look at doing a forward calculation. So what we did here, we just assumed that the root water density, as for example proposed by Fedders, is proportional to the plant water uptake fraction. And if we do this and compare this to the observed deuterium values in the transpired water, we see for the beech that we do a relatively good job in predicting the observed water in the transpiration flux, but we do a really poor job for the beech and oak, and for the oak, where we completely overestimate the isotopic composition in the transpired water. Good. So to summarize this, I think those new laser spectroscopic measurements in combination with the in-situ stable water isotope monitoring technique provide us really detailed insights into short-term infiltration, soil water movement, and soil plant water dynamics. We could find a similar infiltration behavior for all different vegetation types, but we had a higher topsoil and faster event water uptake for the beech. So the beeches in this respect seem well prepared for frequent drought and re-wetting. And as I showed you at the end, that the root distribution alone cannot explain the plant water uptake behavior. So there are probably more physiological properties, or the plasticity very important for that. To end, I hope, despite the short break we had, hopefully in time, I think I have 3 points I will make here. I think we really can do in the future a really smart use of stable isotope to tackle a variety of hydrological processes. I especially believe that sensor networks of continuous in-situ observation of stable water isotopes in the unsaturated and saturated zone can offer really new possibilities in eco-hydrological research, plant research, and soil physical research. And using deuterium 18 oxygen as artificial tracer in the lab and field experiments also will have much more potential in future, since we have those continuous observation in the soil now possible. Good. I would like to thank you for your attention, despite the feedback is always difficult on those webinars, and I thank all the scientists, technicians, and students in my group and in the project for the continuous inspiration and support, and certainly the funding from the German Science Foundation. Thanks a lot.
Thank you, Markus. Now's the time for our question and answer. We have, I guess, about 10 minutes, so that's perfect time. And I guess I don't have any questions that have been texted to me, so I'm not sure if that's because no one has a question or because the text went somewhere else. So if any of the other panelists have one, I guess please speak up or forward to me. But I can get us started with a couple of questions. I'm interested, Markus, in this in your first example where you were estimating the transit times using the isotopes versus the hydraulic modeling. Mm-hmm. And I was really not sure that I understood the difference between the estimates you were getting with the two different methods. Is that difference because they're estimating different transit times, or simply because we're estimating different ways, the same transit time?
No, more or less the differences are that we estimate different soil physical properties in the model, so saturated hydraulic conductivity, water retention characteristics, right? So we certainly use some initial guesses based on the soil texture, but as everybody knows, there's still a lot of variability. So if you just put in those information and we use, for example, on the one side the soil moisture information and try to predict based on the HYDROS model the dynamics in the soil moisture, we get certain soil properties, and we then use those information and we make a forward prediction of transport, and then this may be completely different from if we use, for example, the soil isotope profiles directly in combination with the soil moisture.
Your conference has been extended an additional 25 minutes.
Soil moisture dynamics, and so this can be then linked together, and the estimate of the parameters will be different, which will then again result in a different estimate of the transit time distribution. So again, people in soil science know that if we have a direct use or information from a tracer, and the tracer in this kind is just a natural variation of the stable isotopes, will provide us much better information and hence a better estimate of our parameters. In addition, especially the soil or the stable isotopes help us much more than a conventional solute, since the uptake by the plants is then also related to the stable isotopes that are left in the soil.
Yeah, the reason I ask that is that it seems like, in a way, when we talk about transit times through the soil, we're not often meaning it literally. It's sort of an effective, mean across an unknown parameter distribution. And we recognize that the different pathways are sort of not all completely explicitly represented in the math. And so I'm wondering if you're going to be actually able to tease apart the shape of that distribution using the isotopes, or if we're still looking at a sort of mean effective transit time.
No, in this kind, really, the shape is given by it's still okay. In this respect, you have to believe that Richard model or Richard's equation works for this kind of soils, right? And we can certainly debate about that, if preferential flow may be important for some of those processes. But from the simulations that we did and the forward modeling, that also really means that the shape of the distribution that we get, just an example here, those shapes are really a consequence of the soil properties the water movement and the evapotranspiration or the root water uptake. Right?
I'm still not getting any texted questions, so don't be shy. But I still have more. The next one's easy, which is, you mentioned that you used a different bag than just a Ziploc bag for the equilibrations on your destructive samples.
Mm-hmm.
What's the bag that you used? I'd be interested to.
Yeah, it's actually, we tested different ones. Finally, what we use, it's a little bit of combination of you can certainly get even better bags than we used, but then they are very expensive. So we at the moment get bags that are used for storing coffee beans, you know, the ones where you buy your coffee with. They are really, really gas tight. And since they are used so much in the world for transporting and delivering coffee beans, they are relatively cheap and can be easily used. And so we did some tests in the lab where we just used different bags and left them for weeks and just were looking at seeing how much gas is leaving the bags and do we see any fractionation effects due to that. So if you want to have the company, I can certainly email.
Coffee bags, got it. In a similar, well, actually it's not similar, but in trying to sample the moisture or the vapor from the soil moisture, you talked a little bit about how you were using dilution with the dry nitrogen gas instead of trying to heat the lines to prevent condensation.
Mm-hmm.
And I'm wondering if that's feasible at lower concentrations of water vapor in the analyte gas. In other words, if we're trying to measure, let's say, atmospheric vapor, at which the humidity is presumably much lower than where you're getting it from the soil equilibration. Have you tried it with other vapor sources besides the soil? Are the instruments of sufficient precision to be able to do that?
Yeah. So I think it depends what the other sources are. If you use, for example, as you say, water vapor in the atmosphere, because it's most of the time relatively far from saturation, you should most of the time not have a problem of condensation in your sampling line. However, if maybe in your climate region the water vapor concentration is relatively high, you may have whatever 85-90% humidity, and then you have a small temperature change. There you certainly have that problem. But again, then if you dilute that from 90% to 50% relative saturation, you are still around, depending on the temperature, whatever 10-15,000 parts per million, which is still a very good range for your analyzer. If you do the calibration also for that range, or you do a good correction for your water vapor in your analyzer. But we actually looked at those things and we recognized that if we are not at that upper end and we are well above 5,000, 6,000, 7,000 parts per million, we have no problem in the accuracy of the estimation. If you would go down to whatever 2,000-3,000 parts per million, that would be a range where you probably need to think about the accuracy of it.
And do you never get to those ranges even in the sort of drier end in the soils? Excuse me.
No, that's interesting. People usually think if the soil moisture is low, the vapor content is low, but I think even in soils with only a water content of 5-6%, the vapor or the concentration of water vapor in the soil is still around 90 to 95% relative saturation, right? So always the water vapor you extract from the soil is very high to the saturation, and that's actually then the problem that we mostly have. If you do not dilute, right, you always have condensation, which is the worst thing that can happen, right? Because then you have that water sitting in the lines for a very long time.
And I noticed, do you notice any equilibration time differences in dry soils versus wet soils when you're doing that sampling?
No, not much. So again, we didn't go to 5%. So we looked in what we can usually expect in the soils between, let's say, somewhere in between permanent wilting point and fill capacity and saturation, and there we couldn't see much effects because the porous membranes we use have a relatively high throughput in vapor.
Okay, I want to be not the only one to ask a question. So what I'm going to do, because we don't have too many people on the line, I think we can try this. Let's take it out of lecture mode so that everyone can speak and be heard. And hopefully someone will speak up if you didn't want to text a question. Hopefully someone can have one. So I'm going to do that. Remember that you're now live, and remember to unmute your phone before you speak. So here we go.
The conference is no longer in lecture mode.
Any questions from the world?
Richard, this is Stacy Clark. I sent one to you via chat that was sent to me privately as the host.
Oh, you just got one, okay?
Yeah.
Got it. All right, well, I'll put it back into lecture mode here momentarily then.
The conference is in lecture mode.
Question is, for one of the examples of the sampling in the soils, it looked like there was a downward trend in isotope signature over the course of the experiment. Is that just a sampling effect?
In the last one, we saw that. That's correct. At one location, that was the case. We think it's mostly an effect due to we're probably very close at that location where we have more or less water moving a little bit preferentially along that sensor, where we at the beginning more or less have the water or more of the sprinkling water direct being sampled, and after a while we get a little bit more of that mixture, hence that isotopic composition is being lowered. So we think it's really an observation we made, but we attribute it more to the heterogeneity we have, especially at that topmost sensors that was only 5 centimeters below the soil surface. The soil was very aggregate rich, relatively loose, low bulk density, and there we certainly have some, or may have had some preferential flow close to the sensor at that location. In the deeper ones that was not the case, but that really upper one, there we may have that preferential effect due to the sensor.
Is that downward trend the same trend, or is it opposite from the if it were a sampling effect? In other words, if your extraction of the vapor were causing fractionation, would you be seeing that same trend, or would it be opposite to that?
No. Sorry, what do you mean?
If you were causing some fractionation by the sampling, or some other sampling effect, would the trend be expected to be downward as you saw, or would it be the opposite way?
It certainly would not be as strong because we have such a strong signal. Any fractionation may cause a little bit, whatever, a couple of per mil, but certainly you see we put up 1,700 per mil of our tracer that we applied. So we could certainly even not, if there would be any fractionation, we could not see that. But we tested the sensors for fractionation effects, and we couldn't find any of those up to now. There may be some, doing some more analysis. And so as I'm saying, I really think at that moment water was just moving along the sensor, went close to the sensor membrana, and there was just that exchange then of part of that sensor was with very high concentration, and that there we saw that spike.
Okay, I got one question that's come in right here at the end, but I noticed that it's 1 after. I've got 2 more questions. It's 1 after 12 or 1 after whatever the hour is for you. I want to make sure that we advertise the next 2 sessions quickly, and then we can probably stay on the line. We have a little bit more time, so if you want to hear, if you've got 2 more questions in, then stick around, and we'll do that. Emily, do you have those 2 slides for us? I'm seeing that. I'm not sure if anyone else is.
If you see them, they should be up.
This one?
I see it, but I think, they're up at the top. If If you click on the top, you can see the next event, which is Gabe Bowen talking about fast analysis and flexible networks, citizen science, and hydrologic events. A very interesting talk. And then after that, in 2 weeks, will be Julian Klaus.
Next Wednesday.
Sorry?
It's,
that's right. The next one is on
Monday
Wednesday instead of a Monday. Sorry. Thank you. Don't forget that. I did. We'll talk about high-frequency observations in online measurements in precipitation and stream flow. Okay, back to the last two questions for Markus, though. First, this is for Markus. For soil water isotopes, I wonder if you tried cryogenic vacuum distillation and compared the results with the liquid-vapor equilibration techniques. And have you seen any differences in the isotopic ratios?
We have not done yet, but we are currently running a project where we do that because we do not have a cryogenic extraction line here in our institute. So this we did not, but what we did a lot was compare the isotope, so the soil isotope measurements with more or less soil water samples made with a known source of isotopes. So we just made a mixture of dry soil and dry soil material and water with a known isotope composition. We mixed that, made more or less so our soil prevented any fractionation evaporation, put our sensor in and did the measurement there. So that's more or less what we used and did to track the methods, but I'm saying we did not do any cryogenic extraction up to the moment, or we didn't compare that yet.
Hopefully someone has done that and can present a poster at our poster session. And then the last question is a quick one. How did you efficiently generate your labeled water?
Efficiently. So what we just did, we just bought deuterium oxide and just added that to 1,000 liter of water in a certain concentration. So we got that high average isotope or deuterium concentration, and that's it. So you just need to buy deuterium oxide.
Just buy it.
Just buy it. It's not so expensive anymore, actually.
Okay. I haven't received any more texts. We're 5 minutes past the hour. Thank everyone for paying attention. Thank you very much, Markus. And hopefully we can get some good participation at the following events. Signing off.
Thank you for listening. Thanks, Richard.